Hello students. Today we are going to discuss a topic, very important topic, acid-base balance. Maintenance of acid-base balance is a part of maintenance of human body homeostasis. Human body does a great job in keeping the acid-base balance to normal despite so many biological processes taking place that threaten to disrupt it. It's also significant for a clinician to have a knowledge of acid-base parameters of a patient, particularly lung patients, patients with complaints of lung disease, breathing problems, uh, as there might be a manifestation of acid-base imbalance secondary to an existing disease, and also for a surgeon before the starting of a surgery. We shall be discussing about normal pH, acid-base balance, role of buffers, respiratory system and renal system in maintenance of pH, and finally, various disorders associated with acid-base balance. It will be covered in 3 lecture series with acid-base balance overview in the first lecture and associated disturbances in next 2 lectures. Water and electrolyte balance will be taken up in a different lecture. Before we move ahead, let's just brush up our basics. You already know all these definitions, but before we move ahead, it's imperative to revise them up. So acids are proton donors. Physiologically, the most common acid you will be coming across is H positive or a proton. Bases are proton acceptors. The physiological base is bicarbonate. pH is measure of hydrogen ion concentration, which you know is calculated as negative logarithm of the hydrogen ion concentration designated by this equation. Physiologically, the normal concentration of protons in the arterial blood is 40 nanomoles per liter. It's interesting to note here that protons are in a concentration of 40 nanomoles per liter, such small concentration, whereas other ions, such as sodium and potassium, are measured in terms of millimoles per liter and hence are in higher concentration. And it needs not to be mentioned that it's surprising that even at such low concentration, the protons are capable of influencing the body physiology to such a large extent. So, coming back to the equation, at physiological pH, the concentration is 40 nanomoles per liter. So, if we solve out the pH concentration, pH equation, the physiological pH turns out to be 7.4. Hence, now we know that the physiological pH 7.4 has this term has a mathematical basis. It's not that someone someday woke up and thought that let's set up the physiological pH to be 7.4 on alkaline side as compared to the normal pH, neutral pH of 7.0. Now, buffer, you know, is a solution or a system that resists pH change. So we do expect that a buffer should have both the positive and the negative ions. Isohydria is a term that we use to denote normal acid-base balance. When we are talking about pH and buffers, a brief mention of Henderson-Hasselbalch equation cannot be omitted. You've already learned this equation in your class practicals, and you know that essentially this equation gives about the information regarding pH or pK or the concentration of conjugated base or acid of a particular buffer we are talking about. So how do we define this equation? pH is equal to pK plus logarithm of ratio of conjugated base and its acid. So if we solve this equation out for the most common physiological buffer, bicarbonate, carbonic acid buffer, at pH 7.4, it turns out that the ratio of concentration of the bicarbonate to carbon dioxide is 20:1. Now, What will this equation mean, this ratio mean here according to this equation? If the concentration of either bicarbonate or carbon dioxide increases or decreases, it will lead to change in the pH. And it's also important here to mention that what is important is the ratio of the base and acid, not the absolute values of the base and acid. So the body always tries to maintain the ratio rather than the absolute values of the conjugate base and acid. Apart from theoretical significance, this equation has critical practical implications as well. The acid-base balance is most commonly influenced during breathing issues, lung diseases, during surgery, or it is critical to be maintained at normal if the patient is going to remain on a ventilator. This is where this equation finds its application. The clinician usually requests for the arterial blood gas analysis, which is done by an ABG analyzer. This instrument employs the Henderson-Hasselbalch equation to calculate the arterial blood pH by measuring the various ions constituents influencing it. Coming back to this equation, if there is a primary change in metabolic disturbance, it will lead to the variation in concentration of bicarbonate. Once it is set up for correction, the secondary changes in respiratory compensation for metabolic disturbances will set up and will lead to variation in the concentration of acid. The primary changes in respiratory disturbances will lead to variation in the concentration of acid or carbon dioxide. The correction set up for these primary changes will lead to secondary changes after renal compensation for these disturbances and will influence the concentration of bicarbonate. You should have noticed that in the place of acid, PCO2 is mentioned. The acid carbonic acid of this buffer readily dissolves into water and dissociates into water and carbon dioxide. Carbon dioxide is measured in terms of its partial pressure, and 0.03 remains to be a constant. Thus, if a change in pH is observed, the reason in terms of the respective component can be known, or the value of respective component is suspicious. If the value of respective component is suspicious, the pH variation should be expected. We already know that the normal physiological pH is 7.4. This value is for arterial blood only. The other compartments, interstitial fluid and cellular compartments, have lower pH, a 2.54 low. This lower pH ensures that constantly H positive are being moved down the concentration gradient into the arterial blood. Hence, an absolute value will not be useful. This also emphasizes that the body will have to come up with the methods to maintain this pH to normal. Thus, the range for normal arterial blood pH is between 7.35 and 7.45 units. If there is a fall in pH, it leads to a clinical condition called acidosis. If there is a rise in pH, the condition is known as alkalosis. Although a free fall or an unchecked rise cannot be allowed, as the survival range for a human body is restricted between 6.8 and 8.0 pH units. Human body has devised mechanisms to bring back the pH within the normal range. If there is a disturbance, the whole body system will collapse beyond this range. When we say rise and fall in pH, what does it mean? Or what causes it? A fall in pH is called acidosis. It is due to increased concentration of H positive ions. And what causes the increase in concentration of H positive ions? It's either the accumulation of acids or the loss of bases. Similarly, a rise in pH is called alkalosis. A rise in pH will be due to decreased concentration of H positive. And what will cause the decrease in concentration of H positive? The decrease will be caused by the loss of acids or the accumulation of bases. Why isohydria is significant? By now, we know what is isohydria, that is maintenance of acid-base balance, and how is it disturbed. But why are we even discussing it at the first place? Why is it so important after all? So why do we need a normal acid-base balance? Isohydria ensures delivery of oxygen to the tissues. It is important for the maintenance of protein structure, thus preserving enzyme activity as well. It prevents ionization of metabolic intermediates, thus ensuring smooth running of metabolic processes. It ensures a normal electrolyte balance. It maintains normal muscle contractility, also normal contractility of the myocardium. Thus turns out isohydria is of critical significance. After knowing what is isohydria, why is it important, and having an overview of how it is disturbed, let's now discuss how is it maintained. What are the various organs involved? What mechanism do these organs employ in its maintenance? Near the tissues, concentration of carbon dioxide in the blood is very high. It readily diffuses into the erythrocytes. Here, by the action of carbonic anhydrase enzyme, it is converted to carbonic acid, which further dissociates into H positive and bicarbonate. Because of the production of hydrogen ions, there is a fall in pH, and this stimulates the respiratory center in the brainstem. The center signals increase in the rate of and depth of the respiration. Carbon dioxide is expelled and thus further brings the pH back to normal according to the Henderson-Hasselbalch equation. If the lowering in pH is still not brought to normal by the action of blood buffers or even by respiratory system, renal system comes into action. A prolonged lowering in pH signals kidneys to secrete increased amount of hydrogen ions in order to bring the pH back to normal. Thus, three organ systems, blood buffers, respiratory system, and renal system maintain the isohydria. The body produces more acids than bases. Human body produces great quantities of acid per day through various metabolic processes. Complete oxidation. The carbon skeletons on complete oxidation produce carbon dioxide and water, which again is converted into bicarbonate and H-positive ions. Incomplete oxidation. Incomplete oxidation of carbohydrates gives rise to glucose. Further, pyruvate, lactate, and H-positive are a byproduct. Triacylglycerols form fatty acids and ketone bodies, and again H-positive are a byproduct. Phospholipids give rise to phosphates along with H-positive. Proteins are degraded into amino acids, which also contribute towards sulfate and urea, and yes, hydrogen ions are produced as a byproduct. ATP production is coupled with hydrogen ion production. Thus, it can be seen that human body is evolutionarily capable of handling great amount of acid load. It can be noted here that a vegan meal produces more bases while non-vegan meal produces more acids. The physiological acids produced in the body are of two categories, volatile acid and fixed acid. Volatile acids are produced to the tune of 20,000 milliequivalents per day, while fixed acids only contribute towards 80 milliequivalents per day. Volatile acid is carbonic acid, which is readily converted into carbon dioxide and water. This carbon dioxide, being a gaseous component, is easily expelled by the lungs. Fixed acids, in turn, are constituted by sulfuric acid, formed from sulfur-containing amino acids. Phosphoric acid comes from phosphate of proteins or nucleic acids, uric acid, lactic acid, or ketone bodies. Elimination of fixed acids is not as simple as that of volatile acids. Kidneys employ elaborate mechanisms to eliminate fixed acids produced in excess, which we shall be discussing a little later. We have already discussed overview of the mechanism of maintenance of isohydria. Let's just know in what sequence does it work. The first line of defense against the pH disturbance is chemical buffer system. This is constituted mainly by bicarbonate buffer, phosphate buffer, and protein buffers. Hemoglobin also plays crucial role in buffering the hydrogen ions. While bicarbonate buffer system is active in the blood, phosphate and protein buffer systems are mainly intracellular buffers. If these systems are incapable of bringing the pH to normal, second line of defense comes into action, and that is constituted by physiological changes. The first physiological change is brought in by change in respiratory pattern triggered by the chemoreceptors of the brainstem. If the required effect is still not achieved, the renal system is stimulated to excrete amounts of H-positive ions or bicarbonate ions, whichever is required in the urine. Discuss all these individually in detail, beginning with bicarbonate buffer. It is also known as sodium bicarbonate and carbonic acid buffer. As we know, a buffer has both the acidic and basic components. Hydrogen ions make the acidic component and bicarbonate ions make the basic component. As we've already discussed, a normal ratio of 20:1 of the bicarbonate base and carbonic acid is maintained at all times in the body. The absolute values are 24 millimoles per liter for bicarbonate and 1.2 millimoles per liter for carbonic acid. For the huge amount in which bicarbonate is present in the body, it is also termed as alkali reserve, serving up as an effective mop for the frequently produced hydrogen ions. Second buffer under discussion is phosphate buffer. It is a major intracellular buffer. It is also a urine buffer. Monosodium hydrogen phosphate and disodium hydrogen phosphate are the two major components. The hydrogen ions remain the acidic component, whereas H2PO4 negative and HPO4 negative serve as the conjugate bases. Finally, we come to protein buffers. These include hemoglobin in the RBCs and plasma proteins. Proteins provide buffering action in both blood as well as interstitial fluid. Buffering action of proteins is contributed by carboxyl group, which gives up hydrogen ions, amino group, which accepts the hydrogen ions. Also, sometimes the R groups of amino acids provide buffering by accepting hydrogen or hydroxyl ions. Respiratory mechanism uses mechanical action of lungs. Carbon dioxide, the acidic component, is exhaled. It is rapid and powerful, but only eliminates the volatile acid. The respiratory mechanism has no action on fixed acids. It leads to the adjustment of body pH by changing the rate of respiration and depth of respiration. Renal mechanism is activated as the last resort of pH adjustment and is also the most effective of all. Urine, normally excreted, has acidic pH of about 6. This is due to the large amounts of hydrogens excreted, leading to the formation of acidified urine. In order to compensate for the large amounts of hydrogen ions present, kidneys tend to reabsorb bicarbonate or the alkali reserve so that these excess hydrogen ions can be mopped, and this also ensures the reinstating of the base acid ratio of 20:1. Other methods of getting rid of excess hydrogen ions is excretion of titratable acid and ammonium ions. As the most effective and last resort, kidneys hold utmost importance. If kidneys fail, the pH balance will also collapse. Let's now take up the methods adopted by kidneys in detail. Of the 4 mechanisms just mentioned, the first one is excretion of hydrogen ions. Only kidneys can excrete excess of hydrogen ions, just like the fixed acids. The process takes place in proximal convoluted tubular cells. The action of carbonic anhydrase enzyme converts carbon dioxide and water into carbonic acid. This readily dissociates into bicarbonate and H positive. This H positive ion is secreted into the lumen in exchange of sodium ions, facilitated by ATPase against its concentration gradient. The bicarbonate is transported to plasma along with sodium ions. Thus, the process allows excretion of acid from the body while simultaneously preserving the alkali reserve. Reabsorption of bicarbonate. This process is mainly responsible for the preservation of alkali reserve. Bicarbonate, which is filtered into the tubular lumen, dissociates into its constituents. Na positive is reabsorbed into the tubular cell in exchange of hydrogen ions. These ions combine with bicarbonate produced in the previous step, and carbon dioxide is formed due to the action of carbonic anhydrase present in the membrane of tubular cell. As the amount of carbon dioxide rises, it diffuses into the cell, and in the similar but reverse reaction, the formation of bicarbonate takes place, which is reabsorbed into the plasma. Hydrogen ions again are transported out into the tubular lumen to trap another bicarbonate ion. The Na positive transported in the cell in step number 2 is transported further to plasma by a sodium-potassium antipode system. Thus, there is a net reabsorption of alkali reserve by the end of the process. Titration of filtered buffer. Tubular fluid contains filtered buffer in the form of monosodium dihydrogen phosphate or disodium hydrogen phosphate. You know, phosphate buffer is also known as urine buffer. This dissociates into acid and conjugate base, HPO4 negative or H2PO4 negative. This base is titrated received and titrated by hydrogen ions excreted into the lumen as a result of carbonic anhydrase action as learned previously. The advancing tubular fluid receives more and more hydrogen ions and hence further lowers the pH of urine as per acid load of the body. The bicarbonates produced within the tubular cell from the reaction providing H-positive ions for the titration is reabsorbed into the plasma via interstitial fluid. This capability of buffering of titratable acid gives the knowledge of titratable acidity. Titratable acidity is the measure of acid excreted in the urine by the kidney. It reflects the amount of hydrogen ions excreted into the urine, which resulted in the fall of blood pH from 7.4. Titration of manufactured buffer, the ammonium ions, contributes to elimination of about 2/3 of the body acid load. Ammonia is generated by glutamine metabolism in the tubular cell. It receives the hydrogen ions generated in the other reaction in step 2, catalyzed by carbonic anhydrase of the tubular cells. The ammonium ions thus generated are excreted in the urine. The carbonic anhydrase reaction also generates the bicarbonate, which reverses into the plasma through interstitial fluid. It should be noted that hydrogen ion is always excreted in the urine by ATP-dependent transport system against its concentration gradient. This makes it possible for the kidneys to excrete around 1,000 times more hydrogens than that what is present in blood or cells, thereby substantially enhancing the magnitude of acid elimination by kidneys. A brief roundup to what we have discussed so far. The mechanisms of maintenance of isohydria also act as methods of compensation if there is a disturbance in it. Thus, the three methods of compensation are action of blood buffers, action of respiratory system, and action of renal system. It has to be mentioned here that when students face a question of methods of compensation or role of kidneys in compensation of acid-base balance, they tend to describe the RAS process studied in physiology involving renin-angiotensin-aldosterone system. Although this process is part of correction towards the same effect, it has less significance with biochemistry point of view. The biochemistry of this mechanism involves specific movement of ions, acids and bases across the membranes in specific direction to bring about the designated effect. There is a clear difference in the methods of compensation studied under the physiology and biochemistry, and the two should not be confused in any form. Before we conclude, we should discuss the rates of correction of the three methods of compensation we just learned. Rate of correction refers to the time taken by the particular system to jump into action after the onset of acid-base balance disturbance and bring about correction. Buffer system is the first to respond, acting almost instantaneously. Respiratory system is second to respond, taking several minutes to hours to bring in the required effect. Renal mechanisms are the last ones to set in if the other two methods are unable to bring in correction, although they may take several hours to days. These are the sources from which I've gathered the material for preparing this lecture. I leave you with the cartoon that depicts the various aspects of maintenance of isohydria. I hope you all were able to understand the topic. Thank you. And yes, happy reading.